Oh enggak, anu ya, enggak kedengeran ya, Mas. Pak Al dengar, Pak Al.
Tadi sempat dengar tapi
Oh.
terputus.
Ini pembicaranya Mbak Kinanti nih.
Iya,
Are you Norwegian or Indonesian?
Indonesian working in Norway.
Saya kira tadi eh, sorry, this is English Club ya, apa English anu ya. Tapi saya kira dari India, Mbak Kinanti ini. Ya. Alumni mana Mbak Kinanti?
UGM.
Alumni UGM. Oh, terima kasih Mbak Kinanti. Bener, saya kira tadi pakai bahasa Inggris ini.
Eh, PPT yang disiapin pakai bahasa Inggris
Bahasa
sih.
Inggris ya. Gak tahu ini Mas Raffi pakainya bahasa Inggris atau bahasa Indonesia, Mas? Bahasa Jawa.
Bahasa Inggris, Bro.
Bahasa Inggris.
Global Classroom bahasa Inggris. Nggih, mohon maaf tadi ada kesalahan teknis, izin saya mulai kembali ya, Prof.
Moga-moga.
Nggih, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Thank you once again for joining Global Classroom session with the theme of upcycling food industry side stream into innovative mycoprotein productions. And before we begin, I kindly ask all participants to keep their microphones muted throughout the session to ensure a smooth program. But maybe question and answer will be delivered later after the speaker session and can be sent through the chat box. Without further ado, let us begin for today's program. But for the first agenda is the welcome remarks from the head of the Food Technology Study Program, Mr. Ahmad Nikmatullah Albari, SPTMP PhD. For Mr. Albari, the screen is yours.
Thank you so much for providing me the time for first greeting. Thank you, Mr. Dean, for your attend, and then we are very happy to be with you, Mr. Dean, and then our beloved speaker, Miss Kinanti Mondilaksita, or maybe I can call you Miss Kin. Kin, yeah, hello. Yeah, it is very happy for us to have you here in our global classroom. So we are very happy to receive your information about the upcycling of food industry from side stream into the innovative of mycoprotein production. So it is, I think we need more and more information about this one, since we all of our participants came from food biotechnology. So it means it is completely related with our topic, and then hopefully they can receive the beneficial information from you and in the near future. So this is not only stop until here, so we can continue to another work, and hopefully you still have time with us. Yeah, so Miss Kinanti, and yeah, so yes, we are now in a session of the, facing to the examination, and this is the best time for us to have your speech, and your speech will be our material for examination. So it means this will be good for students to know more and more, not only try to find directly from you, but also from other resources. And thank you also for Mr. Rafli and Ms. Lutfi for your very good management to make this global classroom happen. And I believe that this will be good for not only student, but also for the young lecturer. Yeah, so Miss Kinanti, both of this lecturer is young lecturer, and I also can see other lecturer also join with this class. So I think this is not only student, but also for lecturer as well. Yeah, so thank you so much again for your time with us, and then your information, I believe, important for us, and then we may continue for further collaborative work. Okay, so I think Mr. Dean, would you please to open this global classroom, and then thank you so much again for your attend, and then let me give back the time for you, Mr. Rafli. Thank you so much. Bye-bye.
Thank you, Mr. Albari, for your warm welcome. And we shall now proceed with the welcome remark from the Dean of the Faculty of Animal and Agricultural Sciences, Universitas Diponegoro, Professor Sugiarto, SBT, MSc, PhD. Professor, the screen is yours.
Okay, thank you very much, Dr. Rafli, Dr. Albari, and also Mbak Lutfi, Miss Lutfi, and all our students in the Department of Food Technology, Faculty of Animal and Agricultural Sciences, especially Mbak Kinanti. I suppose that you are from Norway, but honestly, I was so surprised because you are Indonesian. It's very it's honor for us. Thank you very much, Mbak Kinanti. Will share the knowledge from Norway. So this is a very valuable time because we will get a lot of input today. As I know, Mbak Kinanti is working in the industry, in food industry, and Scandinavian countries is very high level in food, especially in food industry. You use kroner, right, Mbak Kinanti? Norway kroner, yeah? I have
Yes.
some Danish kroners until now. So Mbak Kinanti is in Solo. You are in Solo. Oslo or Solo?
Yeah, solo.
Oh, Solo.
But at the moment I'm in Sweden
Oh, in
for
Sweden.
yeah.
I was in Denmark for almost 6 years, Mbak Kinanti. So maybe you can go to Aarhus. You can go to somewhere else in Jutlands. It's very amazing. Anyway, this is a very good time for us to initiate the global collaboration. Be honest, Mbak Kinanti, we need support from the international company to teach our students so that we have a link between university and industry. This is a very good chance for, especially for the student, to get the input, advanced technology, advanced knowledge from the industry. And I believe that the Scandinavian countries, especially in Norway, they have a very advanced technology and very high standard in food. So this is very amazing for us. And not only today, Mbak Kinanti, maybe we will ask you next time when you go back to Indonesia, maybe please come to Semarang and give some insight for our student. And we also need Mbak Kinanti to help us, yeah, to share the knowledge and of course collaboration. Mas Rafli, I think you have a good collaboration. Teman lama ya, teman lama waktu di UGM ya.
Ya, teman
So,
satu PMDSU, Prof.
it's very good because we need also, Mas Rafli, we need peer review for QS World Ranking. So I think we have a very good candidate now. Mbak Kinanti pasti membantulah nanti. Mbak Kinanti will help you and help Undip to increase our ranks in the world. So it is very honorable for us, Mbak Kinanti. Thank you very much.
My
And
pleasure.
tadi disuruh buka sekalian ya? Sorry Mbak
Iya,
Kinanti,
Prof.
because you are
Yeah,
Indonesian,
yes.
so sometimes I'm easier to speak Indonesia than English. Sorry about that.
No worries.
Okay, Bismillahirrahmanirrahim. Undip Global Classroom for the Food Biotechnology class officially open. Thank you very much, Pak Albari, Mas Rafli, Mbak Lutfi, and all of
Thank you
the
so much, Mr. Dean.
teachers who make this class, international class, occurs this afternoon. Once again, thank you, Mbak Kinanti. I officially invite you when you go back to Indonesia. Please come to Semarang.
My
Say hello to
pleasure.
Mas Rafli and us, and give insight for the student. Thank you very much,
Yeah.
Mbak Kinan, and
Yeah.
all of you. Thank you. Assalamualaikum
Thank you.
warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Pak
Okay.
Albari and Pak Sugiharto.
Terima kasih, Mbak Kinanti.
Yeah, for the warm welcome and everything.
Okay, before we move to the main session, it's best for us to take a photo session first. To all the participants, please open your cameras for a moment, and my team will take a picture for documentation. Okay, to all students, please open your camera. Just a moment. Mr. Faris, can you help me?
Okay.
Okay, maybe you can count 1, 2, 3. You already.
Okay, please smile. 3, 2, 1. Okay, once again. Okay, smile. 3, 2, 1. Okay. Okay, Mas.
Okay. Thank you very much for Mr. Abari and Professor Sugiarto for your warm welcome to our speaker. So we have now arrived at the main session of today's global classroom. To facilitate this session, I would like to invite our moderator, Ms. Lutfi Purwitasari, STPMST. So for Ms. Lutfi, the floor is yours.
Okay, thank you, Mr. Rafli. Good afternoon, everyone. Respected Dean of the Faculty of Animal and Agricultural Science, respected Head of the Food Technology Study Program, our honorable speaker, Mrs. Kinanti Mondi Laksita, and respected lecturers and dear students. Welcome to the Global Classroom 2026 organized by the Food Technology Study Program. My name is Lutfi Purwitasari and I will be your moderator for today's session. Okay, so today we are very excited to explore an increasingly important topic in sustainable food system, namely upcycling food industry side stream into innovative mycoprotein production. So, as we know, increasing sustainability challenge in food system have encouraged researcher and industry to explore innovative way to utilize food industry side stream and transform them into high value product. One promising approach is mycoprotein production through fungal fermentation, which combine biotechnology, sustainability, and circular economy principle. Before we begin, I would like to briefly inform you everyone that this session will run until 3:45 p.m. Indonesian time, followed by a discussion and Q&A session. Before that, I'm sorry, Mr. Rafael, for the CV maybe. Can you help me to show me the CV?
Wait, on process.
Okay. Okay, so it is my pleasure to introduce our speaker today, Miss Kinanti Mondi Laksita from Norwegian Mycelium, Norway. Hello, Miss Kinanti. How are you today?
Good.
I hope
Thanks.
you are in a good condition right now, okay?
Yes.
Okay, so Miss Kinanti is a food scientist with extensive experience, in food biotechnology and sustainable food innovation. She completed her PhD in food science from Universitas Gajah Mada and conducted research at University of Borås, Sweden. Her expertise includes side stream polarization, fungi-based protein, circular economy approach, and bridging biotechnology innovation with food industry across international setting. Currently, she works as a food scientist at Norwegian Mycelium, focusing on innovative application of fungi-based protein and sustainable food ingredient. Okay, so without further ado, please join me in welcoming Miss Kinanti Mundi Laksita. The floor is yours, Miss Kinanti.
Thank you, Mrs. Lutfi. I would like to immediately share my presentation first to make sure there's no difficulty, technical difficulty. Okay, so it is shown in the Zoom, right?
Yeah, we can see it clearly, but maybe you can
I
full
will
screen,
start.
maybe?
Yes, I will start my full screen at the moment. Okay, now it is clear.
Not yet.
No. Okay. Wait and
Yeah. 8. Where is it?
nah, okay. But this one.
Okay, yeah, it's
Okay.
clear.
Yep. Thank you very much for having me to attend for this session from Norwegian Mycelium, my company. I'm very glad to share what we are doing here in Norwegian Mycelium to you all, because it's also one of our goal to spread the knowledge like the mycelium, basically. So maybe I can immediately start. Me, myself, I come from the Research Development and Innovation Department. And in my company, I'm the food scientist, but also I'm working a bit in the fermentation scientist part. But it's just to the level of communication between interdisciplinary communication between the team. So I will just proceed. It's a bit laggy. So a little bit of background on why we are doing this. So basically, the population, global population will grow around 33% in 2050. And therefore, the FAO set goal to feed all the population by increasing 60% of food produced in sustainable manner. There is a keyword here, sustainable, by 2050. But due to this sustainability, the expansion of agriculture land is not a viable option. In fact, 90% of deforestation is driven by agriculture. And at the same time, we still have like 33% of food lost or wasted during our food production or even by the end of the consumption. In addition, we also have 17% of water use actually for the agriculture. And therefore, we have some approach regarding this. And I will give the introduction of the company core business. So it was founded by two Norwegian, David and Ingrid. They are my CEO and chief explorer at the moment. So Norwegian Mycelium is a fermentation technology company which transform food industrial side streams or byproducts, if you are more familiar, into high value ingredients using the power of fungi. There's a keyword, mycelium, side stream, and fungi. And at the moment, fungi is a microbes protein source. In fact, why we use fungi is also because there was a study in Nature published in 2022 that stated by only replacing 20% of global consumption of beef at the moment, it could help to halve the deforestation rate. Basically, that's why we go with microbes, microbial protein. And the fungi itself, alongside with its impressive or excellent nutritional profile, They're also geared with very versatile enzyme, and because of that, we can very wide range of substrate from the food industrial side stream, for instance. And then, so now the question is mycelium. It is in our name, but what is actually mycelium? And I would like to introduce it with something that is we are very familiar with, which is the tempeh. But actually, I want to make this presentation quite interactive. But maybe you can send me in a chat if, for instance, do you know what is mycelium, or if you can explain, have you ever think like how to make tempeh and what is that whitish web-like structure that encapsulate or binding the soy protein? So if you could help me to send the chat and discuss it.
Come on, everyone. We can just answer the question from Miss Kinanti from the chat, please. Come on.
It is fine if due to the time restriction. I think I can't continue. If I may, the participants. Okay, so kind of high that you slide the branch of mushroom. Yes, it is the hyphae, right? Any other answer? Thank you, Fergio. And I think I can't continue from that. Is my screen still sharing? I'm just having, okay. So the mycelium, as if we track back on how we made tempeh before actually start the fermentation, there's something that was added, right? We call it either starter or inoculum. This starter or inoculum, we can use some kind of leaf or some that is already powdered, like in the picture. It's actually spores or fragments of mycelium. It's tiny part of the fungi, filamentous fungi, that is later. So if it's cultivated into a correct substrate or suitable substrate, it can start to grow and elongate into that what Fergio said. It's a hyphae. We call it a thread-like structure. And then they, when they are growing more, they communicate, they share by sharing network. They also transport nutrients. And this macro structure that is visible to our eyes is actually the mycelium, and depends on how it is the condition to grow. They can have different type of morphology. It can be very dispersed mycelia that you can see in the tempeh, or more like a clumps that is not too dispersed. And the last one, if it's more often the case when you do submerged fermentation instead of the solid state fermentation, it can clump together to become a pellet. It's like boba, for instance. So, yep, if it's clear, can. Now that we learn about mycelium, I want to zoom out on again our process to produce mycelium, how we produce the mycelium. So first thing first, the substrate in this case, we are choosing a side stream or byproduct that is liquid from a food industry instead of like solid like soybeans. And then we grow the mycelium inside the bioreactors, very controlled condition, until desired quality and quantity is achieved. Then we harvest them. This mycelium, as I mentioned before, they are a network of a nutrition exchange and so on. So naturally or inherently, they are actually containing a lot of good nutrition, but notably is a protein and a fiber as a structure. This mycelium then is the ingredients that we are using to produce hopefully more sustainable food and also sustainable feed. And since the context is in Norway, it's mostly for application in aquaculture. Next. But then I mentioned that we are using bioreactors, we are using submerged fermentation instead of the solid state. We would like to build this solution because submerged state fermentation is actually well established industrial technology, and it is proven to be scaled up to tens of thousands of tons. The second thing is it's quite efficient. By doing shift, we could produce 24/7, and it has a small footprint because it's design of bioreactor that is mostly can go going vertically instead of expand on the area on the land. And from the LCA, it has the carbon footprint of 2 until 3 the maximum kilogram carbon dioxide equivalent per kilogram product, way lower than animals protein, and comparable with some alternative plant protein. It is sustainable because then we have better resource utilization. We use side streams, which is you can almost address it like a waste of the food industry that is still food grade. And because we are using it, it's cost effective and no competition with both food or agricultural land. And the process actually flexible because, for instance, we want to deploy it immediately in the same place with the food industry itself. For instance, it's kind of like wastewater treatment at home. And then we can also utilize the existing infrastructure there. Or we can also tailor it to make the best use of inputs for the given market. A little bit context again about the company. So we have exceptional traction at the moment with the partners in Norway. There is mostly research collaboration through grants with activity heavily on the research development and the scale-up process. It's currently with Sintef and Norge. If I want to put equalization with the Indonesian, it's like BRIN, but it's independent research council in Norway. And then in Japan, we have not only R&D activity at the moment, they are also our main side stream provider is a sugar beet company called Nitten in Hokkaido. And we, at the moment, preparing for a commercial deployment of our technology as soon as in 2026, which is this year. Okay. Tell me if I'm too fast. We move to the next section. Aside from mycelium, I also mentioned a lot about the side stream, but I would elaborate further in this section, like why should it be a side stream? So it's a side stream itself is an immense commercial and impact opportunity that is also come with the challenge itself. Since it is generated from food it's inherently also taking up some good nutrients or fraction of it. which is often actually still food or feed grade. And it has very big volumes. It has a billions liter per year globally. To give you an image, like when you produce 1 kilogram of cheese, you actually end up with around 8 to 9 liters of the liquid whey. That's how big it is. And in addition, you also end up with cleaning in place water from the process and some mixed phase water that you have. It's kind of repetitive, but yeah, it's also nutritious as mentioned because it's the fraction of the food or feed itself. And currently, since it is almost treated as a waste, it's a low cost, almost zero sometimes, because the industry basically just want to get rid of it. And it's too expensive for them to basically upcycle in-house. Okay. And yep, it's underutilized at the moment. So this challenge from industries actually for us is also an opportunity to help to impact to basically from the food byproduct like side stream, we try to recover the nutrition through our technology with the fungi and put it circular back as a food in the food system. However, yep, it's of course not so easy and rainbow and flowery paths. So the side stream itself, it is nutritious, is very complex. But this complexity, beside being nutritious, it also entails that we have basically mix of everything inside. I can give example of whey. It's not only whey, it has also, for instance, the disaccharides such as lactose residue, some minerals, some fatty acid, and protein. So it entails that we need to basically design our process accordingly. Second is it has variability and depends on the season. What it means actually for us, if you see the graphs, it's showing some of the trend, seasonal and random pattern. It's almost never been flat. It's either going up or down or just randomized. What it means to us is like it is not consistent for year round. It's affected by seasonal harvest cycles, raw material variability, like different crops types. For if I take the strawberry from Ketep or strawberry from Bandung, then it will be different, and different suppliers for instance, and then processing conditions of the food itself, the change in how they treat it, they produce cheese, it will affect the composition of the whey, and then storage and transport effects. It really matters for us because it makes our standardization of process quite difficult. And the second one is the one that is most affecting me and my department, or rather our discussion here, is about the product quality, the nutrient recovery, and the process efficiency. And yeah, it entails that we need to design a process quite adaptive. We need to keep monitoring, and we need to have some flexibility in the formulation. Just an example, like when the whey is produced from the cheese, it can vary it in protein, lactose content, depending of the milk source and the season, and whether the cow is happy or not. It's impacting the downstream processing. That's how we design our fermentation and how we concentrate the concentration of the process. Challenge number 3 is again is enormous, but it also means it is low in nutrient density and logistic. And this is the problem actually for the food industry. It's organic, so it has short shelf life. It's to dilute too big in volume to justify transport. or concentration is basically related to the cost to do this all these things. And high production rates, but it's also required rapid disposal. And then we come to like how Anomi is approaching this challenge. Of this, I will share you the theme of our high level process. High level in the means of zoom out process. I will provide two. First is when we produce the feed, just to compare with how we produce the food. In here, after we receive the side streams, we do some formulation to fit with the fermentation needs, and especially to fit the strain or the fungal needs. And then we also do the sterilizations prior to the main cultivation. And then the fungi itself that has been through a seed train, it start to be inoculated. And when we receive again desired concentration on quantity and quality, then prior to the harvest, we did the inactivation through a heat treatment because the fungi need to be inactive. And we employ solid liquid separation to obtain our products separated from the spent medium with lower COD, because the fungi is already taking all the organics, almost not all, maybe some certain degree of the organics. And in the feed case, it went through a drying process and milling. And then we have a final product. It can be for a pellet, or because in the feed case, it's one of the ingredients. So you just mix it with other ingredients. This is the feed case. Enough go to the food case. Actually, most of if the sim or process flow is similar, but then we have downstream that is not only locked to one type of product. It's based on our product portfolio. Then we design this downstream accordingly. It can be in powder or it can be in a wet cake type of biomass. Then again, I would. Is there anybody notice a very different differences between the food and feed case? If you can tell, or if you have any idea, what is the difference from the food and feed case? Could you help me to put in the comment? Or if the lecturer or professor, they can also, if you can, so we can have a discussion. Yes, is for animal. But how is the difference in the process? And if we design food and feed, what could be different? Thank you. Yep. Vergio is, yes, indeed, there is no milling. I didn't put it drying and milling, but that means that the downstream processing can be very different from feed and food. So we can have if extrusion or cooking down here. And the product specification that we have for food and feed is also different. So the process here, all the process will have different conditions. For instance, temperature, long retention time, or the shear stress or something like that. It is different from food case and feed case, even though the big line of production or seam is actually the same. Yeah. Thank you for the participations. And then zoom in into the food case in this, because we will discuss about this rather. The size rim itself is introducing variability, right? And the fungal itself also introducing variability. On top of that, how we design the process is introducing variability. So how do we keep the consistency in product? So we have the basis to do this validation and compliance framework. We have very strict standard operational procedure, just like in other company. And we keep to make sure that we have documented and end-to-end traceability for every process with online monitoring and continuous verification of each step. And we also have system qualifications. So each protocols of the system is hold there. This is actually quite similar when I think you're familiar with HACCP, Hazard Analysis Critical Control Points, and GMP. This is the system that actually inspired. In a way, we still have to apply HACCP. So it's really helped us to design this to keep the quality for the but going into the details of I put some blue lines here onto some process sim. I would just like to give more example on how we do it, and to just give you more concrete image on how we do it. Basically, at the first, the size rim itself, it has so many components on it or nutrition on it. And then we basically, after receive it, we formulate it. But this formulation basically is something that we already optimized. We have golden formulation of type of fungi. And then we follow this, but it's not an easy task, right? Because then you need to analyze, for instance, sugar using HPLC, this and that, using different instruments. However, we actually have, I think I presented, but it was before in summer course. We have a machine learning driven infrared spectroscopy model that basically is like to scan quickly to characterize what is the composition of the size stream. And then it will also help us to guide the formulation. It will give us like, oh, we need to add little bit of, for instance, calcium, or a little bit of nitrogen source, glucose, galactose, something like that. This is how we do this. And then at this point, just like ACCP, we have safety checked. Of course, in this point, like how is there any chemical, biological, or physical visible hazard on it? And as for the biological, we have sterilization control process and also validation, meaning like how much is reduced and so on. It is very important to have the sterilization, not only for safety compliance, it is a must, right? It also can affect how our fungi will grow during the cultivation, and it will affect how is the quality of the fungi. If the fungi is contaminated and not healthy, even though it's not harmful, theoretically not harmful, biological biological contamination, it will still causing the fungi, like for instance, having low protein or having bad structure or something like that. And then on the seed train, we have the quality control of strain quality. The importance of always to check the purity and sterility, meaning that we can do a little bit of sequencing, and some of the tests on the vitality and metabolic figure. So to test how is their performance, is it still okay? And also test on stability, because still it's microorganism. You use it every time, so many times. It can change the stability because it will mutate or something like that. Then we need to check this one too. Then I will put all the upstream and downstream processing together. This is basically just like in other food industry, we have real-time monitoring to keep the condition that we are setting, and sampling to see the performance. And we have strict tolerance or threshold of the quality parameters. For instance, I can give example of this inactivation. In food term, we also need to reduce their Na from the fungi, basically. That is from 8 to 10% to under 2%. And we keep on checking the sterility and also the quality by design. Right? Then is going to the food product development part. So for food product development part, we have all more or less the same generalized pathway on how to develop a food product. But I would like to highlight what is the most important is to know what is your product. You need to characterize it as much as possible. Like can be from the baseline, there is about nutritional or sensory profile to understand the potential. And the second thing is to know like, okay, you know that it has potential, is high in protein, but how will it behave during the formulation? How it will be perceived with the human sensory and so on. So this is actually fall into category of techno-functionality, which include like physical chemicals, mechanicals, and rheology tests. And then this all potential, after you characterize the product and you have end product already in mind, then you try to match it. Either you developed it or you basically create a new product with a strength of your product properties. This is basically the whole idea. And since we are working with not quite so common ingredients, this characterization part is very heavy and is also very important, since it's kind of not like a very common food and little known about this. So yeah, it's step 1, step 2, characterizing, and then step 3 is how do you want to position the product? This is where we actually taken into account from the business department part, because you can say that you want to have a product that is premium or not, maybe specialty like in coffee or chocolate. But then actually from the market it doesn't fit, you don't have the market. So we need to discuss here with and work together with business analysis. And hopefully after we have the product positioning, then it will be easier to pivot into what is the end product target. And then we define what is the product specification of this end product target. And then after that, it is translated into how we process and formulate this food, and we engineer the final matrix. So this is basically the concept and the pathway. There is one really very important part, especially if you produced microbial protein from byproducts of food industry, which is the regulatory. It can be a big roadblock, and it is very important to keep everyone safe, right? So there is a continuous background process behind this to keep that we are regulatory compliance. And then we keep the safe assessment and verification always run concurrently across all of these phases with the help of the regulatory body, like in Norway called Mattilsynet. So it's kind of BPOM maybe, it's like a food safety body. Mm. Okay, then we go to the first part of it, which is the nutritional and sensory characterization. I will immediately jump into example for this one, and in this case, so people can relate maybe quite easily. So here in the picture, we get this mycelium or boba. And then for this, we map out what is the potential, nutrition potential, and just based sensory characterization. We powder it and we analyze it. This is our colorimeters. So basically, for example, in the case of microproteins, it's actually found it contains all the 9 essentials amino acids, and in addition, it has the PDCAAS, basically the score that telling the quality of amino acid is also good. That the score is actually comparable with soy or dairy. So this is potential, right? And the second thing is it contains high dietary fiber, which is actually come from the cell wall. It has beta glucan, which is one of the best fibers that it has potential, then it can help the consumer to meet the recommended 25 to 30 gram daily fiber intake for the gut health. And the third part is actually it has this structure. If you want to make like a meat-like structure, that is actually a nanocomposite. So unlike the meat product that the structure is mostly protein, this fungi is actually have structure that is fiber. It has chitin or chitosan, depends on the type of the fungi, and beta glucan intertwined together. That not only dietary fiber, they're also providing very nice structural integrity. And as for the baseline sensory profiles, we map out that it has inherent taste of umami or earthy, quite neutral aroma, and also neutral color as you see here. It's very important because then we see from here if how it dictates on how we formulate. If the color is for instance a bit brownish or not neutral, maybe like this one, then you might also want to formulate it in a chocolate or curry blocks, for instance. But when the color is neutral, it has like wider range of applications. And then also about the taste, would you use it as umami enhancer, or since for instance it have no bad notes, then you don't have to mask it or with other ingredients. Or since it is neutral, you want to use it to mask other taste or dilute other taste from other ingredient. For instance, if you want to produce from blue mussels that has strong taste, then you want to dilute with this taste without introducing new off flavor. That is the example. The last part is this is also important to be to inform us if we have anti-nutritional components in our fungi. What is very known is it grows fast. It's microbes and it's quite complex microbes compared to other like bacteria or yeast or something. It's inherently actually contain high of RNA. As I mentioned, it's 8 to 10% of its weight. And by mapping out of this, it's part of quality control. Of course, all of the also quality control, and also to check if actually our heat shock procedure is reducing this RNA content, basically. And yeah, I will inform you, but do you perhaps know like why the RNA reduction is important? Can let me know in chat if you know. And why in the food we cannot eat too much RNA. Yeah, maybe it's also quite new, so I can't continue. It's actually a nice information. So basically, the problem is RNA is from purine base, right? And if basically human consume it too much, our digestive system it's breaking it, especially the liver, will convert it into uric acid. And human is very slow at digesting this, and then it will turn into urate crystal. And if it's deposit on the joints, it become a gout. And if it deposit in the kidney, then it's become kidney stone. So this is really one of the safety, the important of this process and why we have to decrease it. Okay, anything else? So we can continue then to the second part is technofunctional properties. You see, this is the microscope image of our fungi is like interconnection of some small myceliums. And in reality, this is the image look like a fibrous chicken shred that you see on the top right. And I will just take a very general approach that we usually do for technofunctional properties that can fit into many types of formulation. So what is usually important is we need to know how is the hydration and binding mechanics, especially the water holding capacity and oil holding capacity, because it's a carrier of the taste and it also dictates the human perceived sensory of the texture, like how hard it is, and also more importantly, the juiciness and the mouthfeel of our product. So most of the time, this is always tested. And the second thing is about how is the matrix formation if we mix it with other things. So we also evaluate the solubility of it, the gelation, and also emulsification index. One of the example of the cases, okay, we have a strong fiber structure, right? But how is natural gelation, for example, how when it's mixed in foods like not surimi, kamabu, fish bowl, for instance. So by measuring these mechanics, you will know how is your ingredient will behave and is it superior or it is actually inferior. And it will help to guide like how to engineer this to have a final bite and chew that is nice. Maybe we need to add binders, that is for example, or we need to add more emulsifier to this. And yeah. You can share your thoughts, can collect this by the end. Okay. So now, for example, as a food scientist, you already know, right? How is the characterizations of your product, then it is time to communicate it with the business part people and to get their feedback and for them to get our feedback. Because in the end, our goal is to sell a product. So the first one is positioning. It is required to basically you need to have cross reference or fact of what is the biological reaction your fungi and then market opportunity. We cross reference like the nutritional strength, high protein, high fiber, and the techno-functional profile with what is in the market, basically what is the need and actually how it can compete. So in order to do that and to help us to navigate this, we do what we call it benchmarking. So we try to compare it against the other common ingredients of the similar position or as the similar role or similar market. It can reveal our mycelium's potential and highlight what to improve or highlight what is the strong point. So I give an example of this table one, the example of benchmarking, the very general one. So for example, we compare the mycelium with the soy protein isolate, SPI, very heavily used in industry, and pea protein isolate is also used in industry, but having like more green label if you compare with soy. So in the structure, because mostly soy isolate and pea protein are in the powdered state, it's amorphous, but we still have the structure of high fibrous hyphae. So maybe this is the plus point. We have exceptional fibrous structure. We communicate with the business analysis, and then it might give us the direction of what is more suitable for end product that can basically compete. And then, but then the question is, we have other water holding and gelation capacity. That is it actually comparable or superior and inferior towards these two? So based on this, it's rather after deciding do we aim for a certain position, the business will sell what is actually a plus point, but then the RDI part will work on how to improve or tailor this potential main main plus point, or how do we make this other functional properties to be at least comparable with the other one. So that is just example of the discussion of the product positioning. And then, okay, for example, we already agree, right, with the business part, and then we discuss and we decide, okay, we decide the product will be premium, so premium position of whole cut chicken breast alternative. So that is the end product. Then we need to know what is the key parameters or the specification of this chicken breast alternative. So we need to define this in order to make sure that, okay, we successfully produced it or we actually totally mimicking it. So this is, for example, what we set up, what is the so we define the human perceptions of the chicken breast. How when you bite it, is it terrible? And then how is the anisotropic muscle fibers behave? Anisotropic is when it will behave differently when the fibers alignment is steered in a different direction. And then after that, we defined the flavor profile. For instance, the savoriness of a chicken, white meat notes with a firm and satisfying chew. And then what is the target of nutrition? In this case, like at least at minimum, it should contain like 50 gram of protein per 100 gram of serving. And last but not least is the target of the cost. Do we aim with it be below, or it's actually on parity or almost the same with the competitors? There's the example, and I can give you the example of the picture. Like this is our mycelium. That we can also label, okay, it has a quite nice fibers alignment, but it's also contain the real fibers, right? So, yep, that's the example. And then, yeah, after that, we translate it into process and formulation. Again, I will put the more general one. It entails, should we, before we create the end product, is there any pretreatment needed? And then after that, is there any further texturization needed after the pretreatment? And then we form the final product. The example of the pretreatment is we can adjust the biomass to basically to be able to accommodate the matrix to for the binders. If we need binders, we can adjust it through mechanical shearing or milling, or we use a pH modulation. For instance, some of binder will bind better in the lower or higher pH. And then afterwards, after we prepare it, we put all together into texturizations. Remember, we need to care about the fiber alignment in case of we want to mimic chicken breast. Like chicken breast, it has a certain direction. We can apply it using high moisture extrusion, or we can do directional freezing. So basically, it's just laying using the fact that the freezing is creating sign of crystal that could help to ice crystal that help to align the fibers. So this also can be done. After texturization, then we use we put them into the final forming. We can mold it and then formulate it into a final consumer ready shape, followed by thermal settings or cooking. So that is the example of the process. Yeah, we can discuss if you have later what type of product you have in mind and what is your plan, or if you have any question, how to process and formulate. This one, I just want to give example of the formulation. For instance, oh, my mycelium apparently needs some binder, so we made up only 75% of inclusion rate of mycelium, but I need to take into account I add binders and oh, I need more flavoring and juicy fillings, and then I add fats on it. And then by the end, apparently it's still less chicken taste, and we can add the flavor, or if the color is need to be more whitish or something. Or even like if it's want to be more like a product that is already shared or not like boiled one. So other product portfolio that can be examples, and we have been trying, and also other companies, our mycelium research about it. It has big areas, but most of the time, or the core category is meat analogs like chicken, beef, and so on, and seafood alternative. It can also be like fish meatballs, or just I think miko sashimi. Some of them I found they tried also to 3D print something like that, and dairy and dessert mimic because it has this umami and creaminess taste. And then egg replacement is maybe not total replacement. From what I read, is more like you can reduce the use of egg because this mycoprotein has the emulsifying capabilities and foaming capabilities. Then you can also use it in bakery, savory snacks, and fillings. Other than that, functional ingredients, maybe you're familiar with some sport food, and when you have high protein bars, something like that, they're usually from sweet whey, not the acid whey. And you can add this microbial protein that also proven to be help with the muscle development. On top of that, you also get the fiber from it. It can also functional ingredients like the fibers. And then it can also be a flavor enhancer because it has umami taste. You can mix it with dashi or stock or something like that. Example. And okay, we'll come to the last part of the product development, which is again is actually the very important one, and you keep doing it in the background anyway. Is first the fact that we are using microbial protein of fungi. We need to make sure that the fungi itself is safe. Some of them you might know in Indonesia is more common for this to produce mycoprotein, right? So there is tempeh that is can be eaten. Is aspergillus that is not safe if you want to eat them. So we need to assess if it's categorized as grass, generally recognized as safe. On top of that, you also need to check whether this is a novel food or non-novel food. So novel food is something that our food that is not consumed, be known to be consumed before 97. In a sense, if your food is novel, is new, not consumed 97, it will direct you to different segment of regulatory pathway. So that is important to assess. So we need to do continuous assessment, and it is not only affected by the microbes or by the fungi itself, but also your side streams and also how you design the process itself. Did you introduce something new? And therefore, we need to keep compile the manufacturing data and then set up formulation safety limits, and we record the historical usage documentation that is required basically by the regulator. In this sense, we apply to the European Union, so it's EFSA. And then they will decide, okay, this is safe, this is not safe, or okay, by the nature of your fungi and your process, then it fall into novel food. Please apply for novel food and it will be like processed in that way. But if it's non-novel food, then okay, just go on and keep your safety and you can produce. So basically that is the example. The second most important, or rather is also very important, is allergenity. And it is actually something that only the food producer can communicate to the consumer. And in fact, like from the data in America, the product that gets turned back is the highest is from the allergenity, and it's quite dangerous because they are really relying on us to communicate what is there. There's no way that they can understand this, and the test itself is quite complex. So we need the constant evaluation of the formulation. Aside from, we also need to test if our fungi that is currently not in the list of allergy in the EU and US, but we need to test if there are any potential of allergy introduced from this, because some people report that they have sensitivity towards microbes. So this need to be tested. And aside from that is something we add, like the side stream or in the process or formulation. For instance, the new binders, if you use wheat or soy, if they are introduced in the formulation step, then the product allergen profile is completely changed. So we need a new risk assessment and we need a new labeling and communication. Basically, that is important. And the last part is toxicity and stability testing. We always run a parallel shelf life study to to understand if it's introduced something toxic and how is the stability, and confirm that any process we use or modification like heat or extrusion, it's not synthesizing a new byproduct that is undesirable or not safe in the finalized matrix. So this is one of the three. Okay, I think I'm reaching really the end of the presentation, but before discussion and question, I have an additional information. I think it's also mentioned by the deans and the head of the department is about collaboration. You have to know me. Currently, we always open for internship. We like for people to have internship. With the duration for a project, at least to really have a project, you need to stay for 3 months. And for the longest is 6 months, because beyond 6 months is like more like a working contract in Norway perspective. And then as for the study background and the major, of course, it's closer to our expertise, like would be ideal, like biotech, nutrition, food science and technology, bioengineering, chemistry and math. But otherwise, really, even you come from other background, take it as you contribute to us rather as an addition of expert in our team. So that also could be nice. If you are interested, just contact us in careers@nomi.no. I provide here. I share the PowerPoint also later, or you just need to contact me immediately at kin@nomi.no. And please visit our website if you want to see more. And then, yep, then question, comment, and feedback session. Yes, thank you. Terima kasih and tusen takk, Mr. Norwegian.
Okay. Thank you very much, Miss Kinan, for the very insightful presentation, especially for the last one about the internship. I think some student will be very excited to try. Okay, so from today's session, we learn a lot. Okay, so that food industry side stream can become valuable research rather than a waste, especially when combined with innovative technologies such as fungal biomass fermentation. And we also learn that developing sustainable protein source require not only technological innovation but also attention to product quality, safety, scalability, and market needs. Okay. Okay, so now we will move to the Q&A session, which will last approximately about, I think we have a lot. And for the participants who would like to ask question, please raise your hand virtually. Anyone? Oh, okay. Aurelino, you can maybe open up your camera and turn on your microphone. Okay,
Okay.
please.
Okay. Thank you, Ms. Lutfi, for the opportunity, and thank you, Ms. Kinanti, for the very insightful explanation. Actually, I'm quite fond of this topic because back then I have I don't know I have an idea about this kind of mycoprotein stuffs, and I'm actually quite interested with this kind of topic. And the thing that I want to ask is, as it stands, Indonesian perspective about the mycoprotein stuff is quite elite, because I think the product like corn in European countries is already usual, right? But the product in Indonesia is considered as a elite or even expensive product. And I've I just searched the product that is near from the kind of mycoprotein stuff is meat analog, but it is derived from soy protein, okay, and not from mycoprotein stuffs. The thing that I want to ask is is it possible to make it as inclusive as it can be in, I mean, considering the Indonesia situation and stuff? Because actually I'm really want to try it, but we don't have any access to it, that kind of a product. And also I see a great potential to turn the waste into that kind of a product. And yeah, the texturization part is also quite expensive, right? Because we have to uh, mix it with binders and stuff. Yeah, is there any concrete or even feasible options to, yeah, to make it come through in this kind of situation in this country? Thank you.
Thank you. Very, very good question. So, yep, the goal is really to be able to produce around the world and make it more inclusive, to be honest. And therefore, yeah, it's common known that it is not super cheap process. But that is also why we are using a side stream to keep the cost low. And inherently, it has already complex nutrition. So really, we don't add too many things during the process. And we have techno-economic study to basically see if this is actually feasible with the range of the price that we are trying to sell, and we aim for the low price already. But of course, yeah, if you sell it in Europe and it's expensive, it's also VAT is quite expensive, and so many things make it expensive. But for instance, we try to work in Japan, or maybe we will also try to work in India, for instance. I think it can be inclusive. The thing is, we will always adjust it to the local context. Meaning that if you build in the facility and so on, we try to sell in a country of interest, right? We want to try to make it also inclusive for that country to be able to for people to buy it. So there should be a marketplace on it. On top of that, maybe for the texturization or something, I was thinking for Indonesian perspective, maybe we don't have to mimic anything, and we are used to tempe. So the production of the mycelium should be more simplified, as it is like as simple as use the thread and then just put seasoning or something like that. But it is just from my perspective. But this is also one of my goal, right? I really want to bring this also into Indonesia, help to secure because it can help to uh, for a country to produce their own protein and from their own food waste, for example, not food waste, maybe byproduct. So, yep. I hope it is quite answering, but at the moment, what I can think of is we can always adjust to locality, and we try to price the cost by using these side streams instead of media, because if you know corn is using hydrolyzed media that is pure from potato, and it's also the main cost of it itself. It could be like 50%, for instance.
Okay, thank you so much for the explanation. Very insightful. Thank you.
Yep, no worries.
Okay, thank you, for the question and the explanation, Miss Kinanti. Okay, so for the next question, anyone? Okay, here, Gio, you can open up your camera. Please. Gio? Yeah, okay.
Okay, thank you for Miss Lutfi for the chance to ask. Uh, so I want to ask about the specification for the side stream because what I know is the quality of the product will be very various based on the material. So how do you make a standard for this product and how to make sure that the quality of the product won't exceed the limit even though it is very various? And the next one is there any some kind of SNE like for this product in Norway or maybe other country? I think that's it for my question.
Yep. Thank you. Another good question. So, yep. Let me just my camera. Okay. Should we proceed with another question, or I can answer this question first, Ms. Lutfi?
Chris, you can answer directly, or you can just collect it. It's
Okay.
your preference.
Maybe I will answer.
Oh,
Belgium.
okay.
Yep.
Then please.
Yep. So basically, in reality, yep, we did a lot of assessment for the side stream itself. Like notably, we samples a lot on how is, and we ask as much as information from the food industry on the variability and so on. So basically, it's data driven decisions. The second thing is also about regulation perspective. That okay, this is intended for food or intended for feed, then accordingly we also put that into dimension. But this data driven thing is to show us like how is the threshold, how is the predicted variability that will we handle, right? And therefore why in the first time we have this called formulation. And basically we have a golden formulation, meaning that this should be followed in order to make it consistent. For instance, it's based on, okay, you should have a certain level of, let's take example, sugar size rim from sugar beet in Tokyo, not Tokyo, sorry, Hokkaido. We locked on the how much sucrose, how much nitrogen source. Basically, it doesn't have to be very similar, but we know a compound that will dramatically alter the product specification, basically. Like I would say the carbon and nitrogen and several minerals because they are kind of accumulating minerals in their body. So based on that, then we formulate it to match this golden standard, and we start to cultivate hopefully the similar strain that we keep always stable. And then we really do a very strict control. We samples the medium. We see how it is the fungal behave. We take sample of both, like the medium and the fungi, like how they are actually following our golden standard or quality that is set. As you mentioned, it's kind of like SNE, but the SNE is by the end of the product. But during the process itself, we have our own kind of threshold or range that we should follow. So that is so far how we do it. And it seems like we can accept many size stream, but at the moment we always a bit focused on what is the size stream of interest. So one design is usually for one size stream. It cannot apply for anything, and we choose what is the size stream that is feasible also for our process. I hope it's helped to answer.
Yeah, thank you.
Yep. Yes.
Okay, thank you, Gio. And the next question from Yosua, please.
Okay, thank you for Ms. Lutfi. And thank you for the insightful presentation and the opportunity, Ms. Kenan. I also interest to this topic, but I also want to know, Ms. Kenan, not the quality or others, but about the impact. If mesothelioma becomes a major source of protein in the future, what impact will this have on livestock farmers who currently rely on animal husbandry and crops farmer who rely on the protein crops such as soybeans and others? Or will mesothelioma not disrupt these two sectors after all? And the second one is regarding the internship. Are there any requirements such as being a last year student or something else, Ms. Kinan? Thank you.
Okay, thanks. Yep. I'm very happy we get a lot of nice interactive questions that is very relevant actually. Yep. So it can be bigger. So the principle is we try not to totally replace, but as a contribution to provide more resource of this, because 60% of global food production is the aim by 2050. And then there is limitation to expand this agriculture or from farmland. So I would say that they can still grow and so on, but then what is our part is to help to provide more protein aside from them, but not totally to replace them. Basically, that is the idea, to keep up with the human growth and also to increase the sovereignty of the protein production and locality. And just then they can produce something that is from here without, for instance, if you do a farmland or something, sometimes you need to import some stuff like soybean meal or gluten meal or something like that. So hopefully this is how we want to contribute. And the second part of the question is there's no strict requirement or something, even though if you come with the background to grow fungi or understand the bioengineering or something, you can always contribute with the project that we can share. So we will share like what is the current project that is related to RDI, and you can choose what is you're interested with, and we will have a training for you at least 2 or 3 weeks. And I think maybe it's just you can communicate in English. Doesn't have to be super fluent, but as long as we can understand each other, it's totally fine. I think so far, yeah, we have students from France and many, yeah, I think mostly from France, for instance. They also use the English as long as we understand, it's totally fine. Yeah.
Okay, thank you, Ms. Kinan, for the information.
No worries.
Okay, thank you, Yeshua, for the question. Okay, any other question for Miss Kinanti here? Anyone? We still have about 15 minutes. About 15 minutes, anyone? Or should I call your name? Anyone? Okay, so if there is no question, okay, Aga. Okay. It's time for Aga. You can. Time is yours.
Okay, thank you, Miss Lutfi, for the chance, and thank you for Miss Kenan for the insightful material. And I want to ask two questions. Based on your experience, what side streams that can give the best result so far? And the second question is, in Indonesia, what side streams that you think it will be good side streams like that? Thank you.
Okay, thank you for the question. That is very curious. So basically, I'm sorry, but I cannot disclose this information. First thing first, because it's related to our IP creation. However, what I can give you an image is currently we work with Nitan Sugar Beet company. So it's for of course it's because there's a case, right? So sugar side streams is currently a good one or feasible one. It's for an example. Of course, I have a lot of things, but I just cannot disclose. But the second thing is about in Indonesia. Of course, we also have sugar canes, molasses, and so on. However, at the moment, it's still competing with bioethanol production, if I'm not mistaken. But in Indonesia, you also have the water from tempe production, and the biggest one is maybe is the pomes. It's kind of oil mill from palm oil industry. Is in terms of the numbers and in terms of the nutrition, it's actually have a potential, but yeah, we need to assess the grade, whether it can go to the food or actually it can be used to just produce feed for chicken, for instance. So that is my yep. But it's a good reflection also for all you guys to think like what could be a possible side stream, for instance, what has potential from industry in Indonesia in terms of the nutrition and also the scale and the need, like what is the emergence of it to be converted into something else. That's really good reflection. Yep.
Thank you so much, Miss.
No worries.
Okay, thank you, Aga, for the question. And then anyone want to ask another question? Maybe for everyone who can join to the Zoom, you can just text the question in the live chat on YouTube. Okay, so for the anyone in the Zoom, maybe any question? Not yet. Okay, so Miss Kinanti, actually, I'm curious for something. Um, Earlier you explained about water holding capacity, oil holding capacity, and the structural properties of mycelium. Um, I'm actually curious whether this characteristic could make mycelium suitable as a carrier material for encapsulation, for example, or for bioactive compound or functional ingredient. And if so, is this capability specific only to certain microbial species or type of mycelium? And that's my question.
Very
Thank you.
nice question, and it's quite inspirative actually. Yeah, maybe this needs to be further investigated, but from my as far as my knowledge, this could be a potential because is the thing is the way mycelium is holding is through its structure of mycelium. So for instance, I saw it in the microscope that it can assimilate the fat globular inside the hyphae. So it can retain it quite nicely. And it can this can be consequently it can maybe used to bioactive compound. But the thing is it depends how they accumulate it in their body, right? We need to see like what is the reason they are accumulating on their body and what is the mechanics. It's simple diffusion or something. Or it can be tailored into a certain fungi that is rather they are eating that material. So they uptake that material into their body, then it's a potential because it remains there. I would tell you that mycelium is quite greedy, I would say. It's, and that's why I love it, right? It's kind of eats many things. But yeah, I think the potential is there, and then you can just freeze dry or something. As long as you keep it's intact inside the cell, it can be potential.
Oh, okay. So the problem is might be actually the mycelium eating that bioactive compound and changing the thing into another compound.
Exactly.
It could be happen. I see, I see. Okay. So anyone, any other question? Maybe from our lecturers, other young lecturers, maybe Mr. Faris or Miss Hasnia, and maybe our PIC, Mr. Kamil. Okay. Any other question? No? Okay, then thank you very much for the explanation, Miss Kinanti. Thank you to everyone for the interesting question and discussion. Unfortunately, it's actually not to limit, but okay. Time limit, we have reached the end of our Q&A session. And before we officially conclude today's session, we would like to present a token of appreciation to our speaker, Miss Kinanti, for sharing her valuable knowledge and insight with us today. So may I invite Mr. Rafli as the PIC of today's global classroom to present the certificate of appreciation to our speaker. Okay, I cannot see your face in here actually, Mr. Rafli. And Mr. Faris, maybe can you help me to take a picture? Is Mr. Faris in here?
Okay, okay.
Okay.
Okay, please. 1, 2, 3. Wait. Wait. Ah, okay. Okay. Thank you.
Okay, thank you, Mr.
Thank you.
Faris. And okay, so once again, thank you very much, Miss Kinanti, for today. Hopefully, today's discussion inspire us to continue exploring innovative and sustainable solution in food technology. Once again, I'm Lutfi Purwitasari, your moderator for today. See you in another global classroom session. Have a wonderful day. Now, I would like to return the session to Mr. Rafli as the master of ceremony. The floor is yours, Mr. Rafli.
Okay, thank you, Ms. Lutfi, and also thank you very much to Ms. Kinanti Muni Laksita or Mbak Kin, the second chance you deliver your materials to us, especially to my student in Universitas Diponegoro. I wish that we can collaborate more further. As our dean said, that when you come to Indonesia, you can visit our faculty, maybe can discuss another research collaboration. And of course, one of our students very interested with the interesting things that you offer in the last slide. So once again, thank you very much to all the participants, to all the students, and to Professor Sugiarto as our dean, Mr. Ahmad Nimatullah Albari as head of the Food Technology Study Program. Thank you very much. We hope that today's session has broadened our perspective on sustainable food innovation and inspire future development in food biotechnology. Thank you once again. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Bye bye everyone. Thank